Clinical Trial Design. I am Dr. Mike Klepper, the President and Founder of Drug Safety Navigator, and I will be today's moderator. The webinar is being recorded for others to listen to later. The link will be posted on Drug Safety Navigator's and EMB's website within a few days. If time allows, questions will be handled at the end of the webinar. If you have any technical problems during the webinar, follow the Citrix GoToWebinar prompts for help. Now, it is my pleasure and privilege to introduce today's speaker, Dr. Michael Mosier. Dr. Mosier is co-founder and director of biostatistics for EMB Statistical Solutions, a data management and statistical contract resource organization. Dr. Mosier is also a professor of statistics in the Department of Mathematics and Statistics at Washburn University. He has served on the editor, excuse me, one moment. Unfortunately, I didn't mute my mobile phone. Taken care of now. My apologies. He has served on the editorial board of the Drug Information Journal and serves as instructor for the DIA Clinical Statistics for non-statisticians and Advanced Clinical Statistics for non-statisticians training courses. Dr. Mosier earned his BS in Mathematics Education and his MS in Mathematics from Emporia State University and his Ph.D. in Statistics from Colorado State University. Dr. Mosier, can you please begin?
Okay, thank you, Mike. I'm glad to see I'm not the only one out there that sometimes forgets to turn my phone off, so I appreciate that. Before I really get going here, I wanted to take a moment and emphasize to everybody that this is only an overview of adaptive designs. It's not going to be filled with a lot of statistical rigor, what I call stat speak. So it's just an intro to what adaptive designs are all about. So hopefully that will whet your appetite enough that you can do further research on your own or consult with statistical companies to help you if you're interested in designing adaptive designs. So to begin, there are a few key points that I'm going to hit as we go through here. The main one is that adaptive designs are really becoming more prominent in the industry right now, and so you're going to be hearing a lot more about them as you move through clinical development programs. So we're going to present some of the common types of adaptive designs and discuss some of the challenges that you'll face if you try to implement these designs. This is intended for professionals in the pharmaceutical industry, and really, as I said, not designed for biostatisticians so much, but just an overview of what you can expect if you take on this task of designing an adaptive trial. So first of all, the $40 question. What is an adaptive design? Well, an adaptive design is a clinical trial design that allows for making changes to the trial as the trial is proceeding. So you can make changes to arms in the trial, you can make changes to the randomization scheme, you can make changes to the statistical procedures, but it's all done after the initiation of the trial, and it's done in a way that does not compromise the validity or the integrity of the trial. Back in 2010, there was a guidance published called a draft guidance for industry by the FDA, and the citation for that is given at the bottom of this slide. And their definition for an adaptive design is a study that includes a prospectively planned opportunity for modification of one or more specified aspects of the study design and hypotheses based on analysis of the data, usually interim data, from subjects in the study. Wow. When you read a sentence like that, it seems a little bit general and a little bit vague. And what it's it's intended to stay general, so there's a lot of things that are covered by that definition. So remember, most trials that we run today are not what we consider to be adaptive. We write a protocol, we do a sample size calculation, we take off and run the trial, and in the end we analyze the data. There's no opportunity along the way to change our mind about anything or to make changes to the trial. So adaptive designs do not necessarily specify the total number of patients that are going to be used. That's allowed to be determined sometimes on the fly. We may not determine which doses or which arms we're going to have in the trial. So what is specified is the scheme for how these decisions will be made. So here are the main types of adaptive design trials. Most of these I will talk more about as I go further on, but just a word or two about each of these. First one says response adaptive randomization designs. And what we mean by response adaptive, well, let me back up. What I mean by adaptive randomization is, say in the beginning of the trial, the first few patients that come in, they have an equal probability of being randomized to any one of the treatment arms. But as the trial goes on, you look at the response rates for each of those treatment arms and have an algorithm set up in advance that will change the probability of the next patient or set of patients being randomized to different treatment arms. So arms that are doing better might have a higher probability of randomization, and arms that are not looking so good, you would lower the probability that patients would end up on that arm. The next one listed is group sequential designs, and these have been around for quite a long time now, and these are your standard designs where you have interim analyses built in and predefined and pre-specified stopping rules. So these are the designs that you've probably heard of, O'Brien-Fleming method or the Lannon-Demetz design. That's group sequential design. A sample size re-estimation design is one where, particularly if you're not real confident about the parameters that went into the sample size calculation, you build in an upfront sort of pilot study. You collect data on a certain number of subjects, and then you re-estimate the sample size based on the parameter estimates from those pilot subjects. So if you, just a real simple example, when you do a sample size calculation, you have to be able to estimate the variability that you expect to see across subjects. And if your estimate of that variability is way too small, then you may end up with a sample size that's way too small at the end. So you do a pilot study, you notice that the variability is a lot higher than you thought it was, so you redo your sample size and add patients to the final total. The next one listed is called drop the loser design, and actually that particular design is named by several different names. I like to say that the drop the loser name is what pessimists call it, and optimists call it play the winner design. But they're really the same design, and they are a specific example of a response adaptive randomization. Adaptive dose finding designs are trials that typically are phase 1 or early phase 2, and you're trying to assess which dose is either the, say, maximum tolerated dose, or you're trying to find the dose that has the highest response rate. Either way, you do a adaptive randomization and assess patients as they go through and end up with one or sometimes a few doses out of a large number of doses that are chosen to go into further development. Adaptive treatment switching designs are kind of similar, except they allow patients, based on interim data, to switch between the arms in the trial. Hypothesis adaptive designs is referring to a more statistical kind of adaptation. The most simple example that I can think of is you may have designed the trial with this possibility for adaptation, but you start out as a superiority trial, so you have an active control group, and at the interim analysis you're allowed to make the decision to switch that to, say, a non-inferiority design. That is a change in the hypotheses being tested. So those are known as hypothesis adaptive design. An adaptive seamless phase 2-3 trial design, these are becoming very popular. At least they're popular in the literature, and they're popular in discussions that people have when they're thinking about what kind of trial to design. And these are nice because the first phase, which is phase 2, uses the adaptive dose finding type design, and you might start with 5 or 8 different doses in this early phase, and then play the winner to choose which dose you're going to move into phase 3 for your confirmatory trial. But it all happens within the same trial, so you don't have to stop one trial, write a clinical study report, recruit new investigators for the phase 3 trial, and ramp up again. It's all done through phases of the same trial, so it can be very efficient in terms of time and expenses. So why would you ever consider doing an adaptive design? Well, there's a lot of reasons. I've got about 3 slides to go through those reasons. One reason is ethical. You can ensure that individuals are not as likely to be exposed to either unsafe or inferior or ineffective treatment regimens. You tend to weed those arms out quickly, so not very many patients end up on those arms. In a traditional type design, you would have to keep, say, a low inferior dose running through the entire trial, and you'd end up with, say, 1/3 of your patients on that. It also allows for a wise use of your resources. If you can decide to stop the trial early, and those resources can be allocated to be used elsewhere, maybe in the next trial or within certain doses in the same trial. And then sometimes we like to have the opportunity to monitor and evaluate the data very frequently so that we can make decisions that are more along the development of that particular compound rather than just specific to that clinical trial. There's administrative reasons for why you might consider doing an adaptive design. I don't know if it's really considered adaptive, but you may want to make sure that the study is being run as it was planned to be, and so you're looking at the data fairly frequently. Some adaptive designs build in the opportunity to correct the population of subjects as you go along, possibly changing eligibility criteria. Make sure that you're hitting the population that your drug is best at treating. And then you can also monitor compliance with the drug and see how well the patients are sticking to the regimen, and then make changes if you need to do that. And then again, I mentioned this before, but you can check assumptions that were used when you first planned the trial. See about the distributions of the parameters that you're going to be trying to estimate, how much variability there is both between subjects and within subjects. You can look at the background incidence rate. Sometimes we design a trial with the idea that if left untreated, or if they get the active control group, that we expect a certain % of response or a certain incidence of adverse events. But as we go into the trial and we do an interim analysis, we realize that in this particular trial, that background rate is very different than we thought. But that has a big impact on the sample size calculation, so that may be an opportunity to adjust the sample size. And then also if the patient accrual rate is very different than what you expected, particularly in oncology type trials where you have a time to event endpoint, the patient accrual rate goes into the sample size calculation, because that affects how much time you're going to get to observe each subject. If the subjects aren't entering the trial as soon as you thought they would, then you're going to have less time to assess those patients, and so that's going to affect your power at the end. There are economic reasons. We've talked about a few of them, but if you can stop the trial early due to either a lack of efficacy or very strong evidence of efficacy, that's going to save a lot of money. If you can abandon a lost cause and put your resources onto the next compound or a different arm in that trial, but you can also do adaptations that help you pursue the most promising either doses or treatment combinations, or sometimes subpopulations. And these sometimes are based on biomarkers, gene expressions, things like that can target populations that the drug seems to be working better in than others. And so that would be an opportunity to adapt the trial. So even if the trial does not stop early, an adaptive trial may take less time than a traditional trial if you gain increased efficiency and can make a decision faster about whether the drug seems to be working well enough to move into the next phase. And for all of these reasons, I think we're going to see that adaptive designs are going to become much more commonplace. Budgets are only getting tighter in the industry, and so that argues for being as efficient as possible. So a little bit of history about adaptive designs. They've really been talked about since the 1940s, and they weren't really implemented back that far, but the statistical literature has examples of adaptive designs from that far back. And yes, you may not have been aware of it, but there is something known as statistical literature. Us statisticians have our own journals that we read and publish in. So some of these designs hit the literature well before they ever actually get put into practice. And part of that is because regulatory agencies tend to prefer to move a little bit slowly and see if things are going to pan out the way that the original authors thought they would. I mentioned earlier the group sequential designs of adaptive trials, and the second bullet here talks about three of the seminal papers: the Pocock paper, which was published in the mid-1970s, and then O'Brien-Fleming quickly after that, and then Land and Demetz further developed the idea of an alpha spending function and applied it to clinical trials with built-in interim analyses. And that all happened from, like I said, about 1975 through the end of the last century. We have a new design now known as continual reassessment, or continual reassessment of subjects as they go through the trial. You look at each patient as they finish their part of the trial and make decisions about what you're going to do after that. Well, those were kind of developed in the 1990s. Then back in 2004, the FDA began what was known as the Strategic Path Initiative, which talked about a lot of things besides adaptive designs, but it included a fairly intensive discussion about adaptive designs and what should happen in that arena. And I think to their credit, the Food and Drug Administration, as well as other regulatory agencies, have embraced the idea of adaptive designs and are very interested in seeing the most efficient use of patients and resources, which is in the interest of both regulatory agencies and the industry. And then I've already mentioned that in 2010, the FDA issued a draft guidance on adaptive trial designs, and there's a lot of good information in that guidance. So let's talk about a sample size recalculation design. This is one of the simpler types of adaptive designs. but I've already said most of what's on this slide, so I'll kind of hurry through it. But sample sizes in the beginning are what I like to say a spooky thing. It's kind of scary sometimes to set sample sizes based on estimates of what clinicians together with statisticians think is probably going to happen. Well, there are times when you really don't have very good information about the variability or about the response rate or about what would be expected to happen on your new treatment. And so building in this internal pilot study at the beginning of a larger trial with the opportunity to recompute the sample size and just either confirm that you got it pretty close in the beginning, or if you were vastly off, you can make changes to it. There have been some papers published in the last 10 years about this type of design that have shown that by doing sample size recalculation, you actually can affect the type 1 error rate in the trial. And there's some stat speak, which I promise not to use, but type 1 error rate simply means that the probability of getting a false positive, of deciding a treatment works when it really does not work. And so there is some inflation to that. So as long as you do these designs with that in mind and make adjustments for that, then they can be statistically valid. But if at all possible, you need to maintain the blinding of the study to do this. Now, I put this thing down at the bottom that says get regulatory approval. Really, any adaptive design that you're considering, you're going to want to talk to the regulatory authorities and get their buy-in on your ideas before you take off and run the trial. But I put this in from personal experience. We were designing within our company a sample size recalculation trial, and when we presented it to the FDA, they had some issues with how we were adjusting for our type 1 alpha. So it was better that we knew that up front and could change it to their satisfaction than if we had waited to the end. So if we can use blinded data and not risk the validity of the trial by doing any kind of unblinding, then what we're going to do is we're going to base any new sample size calculation only on estimates of parameters which control the variance between subjects and not on observed responses or treatment differences between the treatment. Responses and response rates should be pooled across the treatment groups. So you have an idea of the overall response rate. Or if you need to look at differences between treatments, then you need to have schemes in place, well documented, that explain how we're going to allow some people to know and we're going to keep other people's from not knowing. Did I just say other people's? Sorry about that. If you do use unblinded data, then that puts the trial at a little more risk of validity issues. But sometimes we can only evaluate the validity of the assumptions that went into the sample size calculation once we unblind the data and see what the difference is between the treatment groups. So if we can build methods and procedures into the plans up front to deal with any biases that could creep in, such as using an independent data monitoring board, then we can adjust the sample size at interim analyses without so much risk. So down at the bottom, there's a list of a few options that can be done once you look at unblinded data: adjust the sample size, drop the loser, or adapt the randomization to play the winner, or another type of design known as enrichment designs allows for the enrichment of population. So if at the interim you notice that there's a subgroup of patients that seems to be responding the way we expected, but others are not, you might decide to change the entry criteria for the trial and focus only on that subgroup. while that may not be a direct change to the sample size, it is a direct change to the number of patients in a particular promising subgroup. So we do consider that a sample size adjustment as well. So by adjusting the sample size, if we base it on the effect that we've seen at the interim, then often this is done in a couple of ways. I need to pause here for a minute and explain. I have the second and third bullet says frequentist and then Bayesian. And these are terms that you may have heard, but there's basically two camps of statisticians. The traditionalists and the way that I was trained are called frequentists, and frequentists would compute what is known as conditional power. And what that is the probability that based on what you see at the interim, what's the probability that the trial is successful at the end? So you make the assumption that the observed treatment differences, the observed variability, all of those parameters are going to be exactly what you see at the interim, and then from there you do a power calculation to see what's the chance of success at the end of the trial. The Bayesian approach is similar to that, but it's kind of done in the opposite direction. They don't call it conditional power. What Bayesians refer to it as predictive probability of success. And it's done in a way that assumes, instead of assuming that the parameters you observe at the interim are fixed quantities, it assumes that there's some distribution for those unknown parameters, and those parameters themselves can be thought of as random variables. But the idea is the same. You're computing the probability that the trial is going to be successful, meaning it will have a statistically significant difference between treatment groups at the end. Then based on the results of that calculation, we compute a new sample size that keeps either the conditional power or the predictive probability of success at a sufficiently high rate. So in terms of power, we typically power at 80% or 90%. So we want to keep that level pretty high. There's been some papers published in the statistical literature in the last 5 years that suggest that in the case of conditional power at least, that you may not even have to adjust your alpha for the trial, that type 1 error rate, if the level of conditional power at the interim is sufficiently high. So in other words, if you didn't lose a lot of power from what you expected, then your alpha is going to be okay in the end. So let's talk about the drop the loser design, or as optimists would say, play the winner design. The basic concept is that you start with a multiple armed trial. So you have sometimes what I would consider mini arms, 5 or 8 different treatment arms. And then each of those arms is compared to a control group at pre-specified interim time points. And then at each of those interims, you reassess the chance of success for a particular arm. And if the arm is not doing well, you may drop that arm. Or if that arm is having a much higher incidence of safety concerns than you expected, then you may drop that arm. The benefit of doing this is that it conserves patient resources. Patients that are eligible to enter the trial can now be focused only on the few remaining doses or arms, rather than being spread across arms that are not doing well. There is a concern of type 1 error inflation. It's possible that an arm is looking really good at an interim just by random chance. You just happen to get some subjects in that arm that did really, really well. And so now you're going to focus in on that arm, put a whole bunch of patients on it, and so you essentially become overpowered on that arm, so it increases the chance that you make a false positive statement about that particular dose. So we build into it that that there will be an adjustment to alpha to correct that. But if alpha is not reallocated from the dropped arm, then overall the alpha is not affected. And again, that's kind of some stat speak, but if you think of multiple comparisons, and each of the comparisons, each of the doses is allocated a certain amount of the overall alpha level, then if you don't, if you drop an arm, that alpha goes away, then you can build it in so that your overall type 1 error rate is not going to be affected. So one type of phase 1 trial that we are all commonly faced with are known as single ascending dose trials or multiple ascending dose trials, shortened as SAD and MAD trials, which I always thought was kind of funny. And the typical design for those kind of trials were what is known as a cohort design. Often it's a 3+3 cohort design. And in short, what this means is that you have sequential cohorts of, say, 3 subjects. You start the first cohort at a very low dose, and as long as nobody experiences any safety concerns, then you recruit the next 3 subjects and give them a higher dose. Well, simulations, computer simulations of these kinds of trials have shown that that type of design can behave poorly, meaning that it has a fairly high chance of focusing in on the wrong dose to be the maximum tolerated dose. Instead, Bayesian principles have been applied to this type of design that allows to update the knowledge of the relationship between what dose you're giving and its probability of a toxicity. What statisticians would say its distribution across the doses. And as each patient finishes, you say, okay, this patient got this dose, this patient did not have an adverse event, so that dose, there's a higher chance now that we consider that dose to be safe, and so you make an adjustment to which dose the next patient gets based on what happened in the last patient. So it's called continual reassessment because you're literally doing an interim analysis after every single patient. Obviously, this is not going to work well with a clinical endpoint where you have to wait a while before you know what the response is going to be. But you can know very quickly when you give a single dose to a healthy volunteer whether they show any adverse reaction or not. So it works well in that situation. So some advantages: the patients are always treated at doses that have toxicity rates closest to the desired level. And that sounds kind of funny. When I wrote that bullet, I thought, toxicity rate that's desired? But what we want to do is we want to control the toxicity rate. We know there's going to be some kinds of toxicity, but we want to keep the rate fairly low. So what we want to do is find the maximum tolerated dose, and that's defined as one that has a toxicity rate that's acceptably small. In the end, computer simulations have shown that this type of design does better overall in terms of number of patients. You're able to determine more accurately the maximum tolerated dose and do it with fewer patients, on average fewer patients than you would get in the 3+3 type cohort design. So a response adaptive randomization trial is one that changes the randomization to the different treatment arms based on how the subjects are responding. So you may start the trial with, let's talk about a 1-to-1, a 2-arm trial. You start out with a 1-to-1 randomization ratio between the groups at the beginning, but it randomly assigns a higher proportion of patients to the arm that is doing better. So at each interim look, you adjust the probability based on the response rates that are seen in the trial. These are also known as outcome adaptive randomization trials. And they have a very intuitive appeal because on average, a higher proportion of the patients will be treated using the better or the most promising of the treatments, if there is one. If you're not seeing any difference between the treatments, then it stays roughly at 1-to-1, and in the end you say, well, I can't distinguish between these doses. There's many of these type of designs out there, but they all share a common theme. And that theme is that we're going to consider many options. Those may be options of doses, they may be options of treatment combinations, or they may be options of patient subgroups that we're going to recruit into the trial. But they all start with many options and then use interim looks at the data to see response rates in those different options, and then typically use Bayesian probability algorithms to change the probability of future patients being randomized into that particular option. Arms that are doing poorly will eventually stop enrolling altogether, and arms that are doing well may do what we call graduate to the next phase, or at least continue enrolling in that arm until the results become statistically significant and conclusive. So here's a quick example. Suppose we start with 5 arms in a trial. Oh, I just noticed it says 6-arm trial at the top, and then I only have 5. I must have ran out of room graphically. So as you start, you go into the screening process. At the beginning of the trial, the adaptive randomization is set so it's equal probability on each of the arms. But as you proceed through the trial, you run your first interim analysis, you notice that treatment arm 2 has not had any responders, and so it gets dropped for futility. The probability for allocating to that arm becomes below some predefined threshold, so it's just completely dropped. Then at the next interim, arm 5 has met a threshold for sufficient number of responses and makes it into the next phase. So enrollment in the trial, enrollment into that arm in this trial then stops. So one has been dropped because it was futile. The other one has been dropped because, okay, we have the information we need on that arm. We're going to graduate it on to the next phase. And then maybe arm 3 graduates, and so on. And you continue doing this until you can make a decision about all 5 arms. So in the end, when you begin to design an adaptive trial, there's different things you need to keep in mind. And I will say that I've designed many trials in my career, and I get the question fairly frequently nowadays that, you know, what percentage of the trials that you see out there are adaptive? And my answer is that, unfortunately, I've designed quite a few more adaptive trials than we've ever actually implemented, because in the end, oftentimes the sponsor chickens out. They go through the whole process of investigating the idea of an adaptive design, but then they become afraid that, well, that's kind of new, it's not what I'm used to, so they don't go ahead and do it. So, and there's valid reasons to not do it. So I'm not saying that that's a mistake necessarily, but there's certain goals that you want to keep in mind with what you're trying to accomplish with your trial. Don't attempt to answer too many questions with a design that's going to be adaptive. Adaptive trials are really good at focusing in on one or a few things, but they're not going to be good at assessing a laundry list of 10 secondary endpoints. Use statistical simulations to understand and adjust the characteristics. So the statistician involved with designing the trial will write programs that will use the assumptions at the beginning of the trial to simulate, well, if we get this many patients in, and as we proceed, we do an interim, and can run 1 million hypothetical trials and see what percent of those trials are actually successful if you make certain assumptions or if you do certain adaptations during the trial. So the use of computer simulations is very critical to the design of adaptive trials. But also you need to seriously consider the logistics of conducting the trial. You need to have a DSMB in place, a data safety monitoring board. There's logistical considerations with that. If you're going to do fairly frequent interim analyses, that involves a lot of logistics. It's hard to get those interim analyses, get the data cut off, get the data entered and locked, and analyses run. So there's a lot of complications to running adaptive designs that you might not hit in a more traditional design. So when should you consider doing an adaptive trial? Well, they're really only going to work well. These are particularly the adaptive randomization type designs. They're going to work the best if it's possible to run interim analyses pretty frequently, say even monthly, and decisions can be made very quickly. So the response needs to be something that can be assessed rapidly. So you're not going to do an adaptive randomization trial in overall survival. It just takes too long to see the response. If you have great uncertainty about the design of the parameters when you do your power calculation and sample size calculation, then you may want to consider adaptive randomization that allows you to reassess that. If there's a really high cost and or high risk for the patients involved, or you have a limited trial population, then you might want to consider adaptive designs because they've been shown to be more efficient use of resources. But again, it has to meet all of the above conditions as well in order for it to be considered. So, quick summary. Adaptive designs are becoming in vogue. You hear a lot about them anymore. As I say, some sponsors are still hesitant to implement them, but I think they're now at least willing to listen and kind of go through the process of seriously considering it as an option. And so you're hearing a lot more about them as we go. They can be more difficult to run those trials, but in the end, the benefits may outweigh that headache. So the comfort level is growing, but it's not. It's still maybe not where we'd like it to be, but I think we're going to get there. The last bullet says get buy-in. As I said before, make sure that you always are very upfront with regulatory agencies that are going to be reviewing the trial in the end to make sure that they're comfortable with your ideas on the design. Okay, so kind of wrap up, we've discussed what the common types of adaptive designs are and some of the challenges that they pose. So just be aware that you're gonna be encountering these more and more as you go through your career. So I will let Mike Klepper take some questions, and my email address is here if you're interested in asking questions offline or have any follow-up.
Dr. Mosier, Mike, thank you for this very interesting and seminal presentation. We do have some time for some questions, and I would like to ask you the following. How can trial adaptations bias the trial?
Well, that's a great question. We've I talked quite a bit as I went through there about trial integrity and trial validity, and that's what I'm referring to is the bias. And bias can creep into these designs in several ways. It depends on what's being changed or adapted in the adaptive design. The simplest example, think of the traditional group sequential design with stopping rules. That is a type of adaptive design. If the results of the first interim show none or little treatment difference, and that becomes widely publicized to the investigators, they may decide to change who they're willing to enroll in the trial. So you've actually had a shift in your target population that's going to bias the trial either for or against your compound, and it's hard to know which. If you do a sample size re-estimation, the observed treatment difference at the interim could be simply due to random chance. So if we base a new power calculation and re-estimate the sample size on a random occurrence that really isn't representative of the truth, that's going to bias the study and possibly be misleading. So in general, from a regulatory point of view, the use of adaptive designs may introduce operational bias, such as selection bias or bias that results from the method that we evaluate the subjects, early withdrawal, modification of treatments. And so because of these things, it may not be able to control the overall type 1 error rate at the levels that we think it is. And that's a statistical bias.
Thanks, Mike. Let me ask you another question. What is meant by an enrichment design?
Okay, and I mentioned enrichment designs a minute ago. There's actually an FDA guidance document on enrichment designs. In short, it means using trial information to narrow the patient population, say via selection criteria or biomarkers or something like that, to make it more likely that the patients will experience an effect of the treatment. So you try to narrow your patient population down to only those that are going to respond. You're enriching the patient population. That guidance has this quote: "For the purposes of this guidance, the term enrichment is defined as the prospective use of any patient characteristic to select a patient population in which detection of a drug effect, if one is in fact present, is more likely than it would be in an unselected population."
Okay, another question, if we can. When you say adaptive designs often use Bayesian principles, what does that mean?
Yeah, I will come clean and say that my training was as a frequentist, and so I've been a slow comer to the Bayesian side of things. The statisticians joke it's coming over to the dark side. But basically, the whole idea revolves around conditional probabilities. So as statisticians, we constantly deal with what are called conditional probabilities. So what this simply means is like, what is the chance of event A happening given that we know event B is true? The usual p-value that we compute is this kind of a probability. It's a conditional probability. What is the chance of getting data like this given that the null hypothesis is true? Well, I always joke that that's backwards of how our minds work. The way our minds work, we really want to say that the other way around. What is the chance the null is true given the data we've observed? So the event and the condition get reversed. Well, Bayesian principles are a way of reversing that conditioning. So instead of considering the null value, the null hypothesis of some population characteristic, let's say the mean, instead of assuming that it's some fixed value, we now assume it also is random and it has some kind of a prior distribution. Then based on the observed data, we can update the likelihood of any given value of the population mean and assign new probabilities. This is known as the posterior distribution. So that's the idea behind Bayesian principles. So Bayesian principles in an adaptive randomization scheme would say, well, the response rates at each of these doses are not fixed, but they follow some distribution. So we're going to use the current date to update what we think that distribution is, and once we're comfortable that we've zeroed in on the most likely candidate, then we can stop.
One last question for you. What is predictive probability of success?
Yeah, I think I threw that term out earlier just kind of in passing, but predictive probability of success, again, it comes from the Bayesian side of things. In short, what that is the probability of observing a success in the future based on existing data that we can see now at an interim analysis. So conditional power is similar, but done from a frequentist approach, meaning that we assume some fixed value of the population parameter, and if we're doing it at a sample size re-estimation, then we're making an assumption that what we just observed at the interim is actually the population truth. Bayesians argue, well, that's never going to be the case. So it's basically the same thing, but from the Bayesian perspective of turning it around. It assumes some distribution for the population parameter and then uses those interim results to update this prior and compute the probability of a statistically significant difference at the end of the trial based on the data we've collected up through the current interim analysis.
Well, Dr. Mosier, I want to thank you again for this very interesting presentation, and I would like to thank the attendees for attending this webinar. This now concludes our webinar, and we hope you enjoyed it.